in the cardiovascular system, we talk about the blood, the heart, and the blood vessels. The heart most of the time steals the attention, and we kind of forget about the functions of the blood vessels. They're there transporting blood that's pumped by the heart and deliver it to the tissues, and then collect blood from the tissues and return it back to the heart. During this process, blood vessels are abused to some extent, because imagine how blood is forced on the blood vessel wall, and then the force may cause damage to the blood vessel wall. And sometimes the blood vessels are twisted, coiled, and blood has to go through these types of channels that are created, and so on. So it's an interesting structure, it's an interesting component of the cardiovascular system to be talking about. So some other interesting facts to consider here is that the length of the vessel, okay, if you were to take all of the blood vessels in the body, lay them next to one another, the distance they occupy is around 60,000 miles. Approximately 25,000 miles is when you can go around the world, and now 60,000 miles tells you that you can go around the world more than 2 times. All right, and then the function of these vessels is to transport 1 million barrels of blood. So that's spectacular, isn't it? How much work they do to support the body. The number of the large vessels like the arteries and veins is fewer. You're talking about hundreds of arteries or veins. These are mainly the big ones, not the branches you're talking about. Then arterioles can be in hundreds of thousands, 500,000 arterioles, and look at the number of capillaries. They make up the most, 10 billion capillaries. So there are more interesting facts that we talk throughout the discussion. And so let's now talk about the anatomy, the arrangement of blood vessels. They form 2 closed circuits. 2 closed circuits. One of them is the pulmonary circuit. It's a smaller one than the systemic circuit, the main one. Okay. The interesting thing is when the heart pumps blood to its blood vessels, whatever blood vessels that are connected to the heart, then the same amount of blood is released into both circuits. Same amount of blood. So you're talking about approximately 5 liters of blood into pulmonary circuit from the right ventricle, and approximately 5 liters of blood into the systemic circuit from the left ventricle within each minute. Okay, so in each minute, you're pumping about 5 liters of blood here into the pulmonary circuit, which takes blood into the lungs. So it receives 100% of the blood that is ejected by the heart, And similarly, the other 100% of the blood is taken here into the systemic circuit, out of which major organs that receive blood are shown here: digestive system, approximately 21%, kidneys, 20%, the brain, 15%, skeletal muscle, 15%, skin, about 10%, and so on. All right, so now we need to talk about yet another important detail, that is the types of blood vessels. Types of blood vessels. The blood vessel that originates at the heart, all right, starts at the heart and leaves the heart. Leaves the heart is what you call as an artery. One of them that you see here that's shown in red is the aorta that starts from the left ventricle. The one that is shown in the blue is the one that starts from the right ventricle, and this is what you call as pulmonary artery. Okay, so pulmonary artery here from the right ventricle, aorta from the left ventricle. So these are the two arteries. Interestingly, what you're seeing here in the blue is deoxygenated blood. Pulmonary trunk, pulmonary artery carries deoxygenated blood into the lungs. And the lungs are the ones that oxygenate the blood, and then the oxygenated blood is shown in the red, and that is the one that arrives at the left atrium as pulmonary veins. There are 4 pulmonary veins, and anyway, so 2 from the right, 2 from the left. That's not all shown here, just assume that this is a combination of 4 pulmonary veins. Oxygenated blood arrives at the left atrium and then into the left ventricle and then out into the aorta. Aorta is the one that supplies blood to the rest of the body, including the lungs. Lungs also receive a branch from the aorta, okay? A bronchial artery is how you call that. So in any case, you got the 2 circuits, pulmonary and systemic, and then the volume of blood they each receive, and then the names of the vessels that originate, and the blood vessel that brings blood back to the heart, the pulmonary veins. So now, yet another thing to talk about is the different types of vessels. Well, we talked about the artery already. The aorta, for example, that starts here at the heart, as it leaves the heart and as it moves further away from the heart, it begins to branch. There are a number of branches, not all are shown here. This is a simplified version just to give you the big picture. So the aorta branches and gives off mid-sized vessels, smaller arteries, and so on. All right, so as they branch and become smaller and smaller, the smaller arteries are called arterioles. Arterioles, again, not shown here specifically. Arterioles branch further and further and then give rise to the smallest vessels called capillaries. What you're looking at here is a capillary bed or a capillary network, okay? So here the color is purple, and the purple color tells you that it is a mixture of oxygenated blood and then the deoxygenated blood. What happens is the oxygen that is brought to the capillaries is delivered to the tissue, and now the capillaries that leave the tissue are collecting deoxygenated blood. So it's a mixture of oxygenated, deoxygenated blood. And then as they leave, it would be mostly deoxygenated blood, and that's why the color is blue. Now you're talking about smaller veins formed from the fusion of the capillaries called venules. Venules are not shown here. You just have to assume again. So venules formed by the fusion of capillaries come together to become larger vessels. As they fuse again, they would be called mid-sized veins. They fuse to become larger veins. Two large veins that you could talk about right away is the inferior vena cava here, and then the superior vena cava. So that's not broken down here, unfortunately. Vena cava is basically mentioning both together. All right, superior and inferior vena cava are the two large veins. They empty deoxygenated blood into the right atrium, and from there into the right ventricle, and then out through the pulmonary artery. Okay, so we talk about artery branching off to become smaller arteriole, which then branch further to become capillaries. Capillaries fuse to become venules. Venules continue to fuse to become mid-sized veins, and then ultimately large veins. So with this, we would be talking about the types of blood vessels in detail here. This is where you're looking at the histology of the different types of vessels. As I mentioned, arteries becoming arterioles, and then capillaries, capillaries becoming venules, and then veins. Okay, and sometimes, the myth is that arteries carry oxygenated blood. Is that true? No. The reason being, there are arteries, like pulmonary arteries, that carry deoxygenated blood to the lungs. Okay, so that's where it's not correct. Arteries can carry both oxygenated and deoxygenated blood. So the best definition is to say arteries are the ones that carry blood away from the heart. Veins are the ones that return blood back to the heart. So that is how you should define, and then the branches get different names in between. Okay, very good. So what you're looking at here is two types of arteries. Elastic artery, as the name suggests, would have a lot of elastic fibers compared to the muscular artery that has fewer elastic fibers. It has elastic fibers, but not as many. So the wall looks a little thinner, and there is more muscle in it. All right. So elastic arteries, interestingly, can stretch. And when the blood is ejected, they can stretch, and then as the blood moves further down, they can recoil. Okay, by being able to do that, stretching and recoiling, they can be the pressure reservoirs. When they recoil, there is that bouncing type of effect on the blood. It pushes it further. All right, so that is nice. Now let's talk about the histology a little bit closely. The blood vessel wall can be made of three distinct layers: tunica intima or tunica interna, tunica media, middle layer, the tunica externa, also called as tunica adventitia. Okay, those are typical layers in a vessel that's large and exhibits all three layers. This is not true of a capillary. Capillary is made of just one layer, that is tunica intima. You have one layer of cells and then the basement membrane. Okay. Anyway, that leads us to talk about what is in each layer. Tunica intima is made of a layer of cells. These cells are very important for us to talk about. These are called endothelial cells. Endothelial cells are the innermost layer of cells. They are the ones that come in contact with blood. They are able to secrete a variety of substances. Now, because they're in contact with blood and because blood can be rushing through the vessel, damage to this vessel, it can be quite common. Okay, injury to the vessel, injury to the cells can cause inflammation. Inflammation can lead to release of more chemicals that could lead to long-term complications like atherosclerosis, etc. So we will be talking about endothelial cells in context with many conditions in the different places. Anyway, so tunica interna, tunica intima consists of endothelial cells and a thin layer around it that would be called a basement membrane. All right. Then you have tunica media. Here you have some loose connective tissue and some smooth muscle fibers. Okay. So here is where you also see elastic fibers blended with the smooth muscle fibers. Okay, so loose connective tissue has collagen fibers, elastic fibers, and so on. So the amount of these fibers vary depending upon the nature of the vessel. All right, tunica externa is more or less a connective tissue layer. It's the outermost layer, and that is tunica externa. Now, the muscular artery that you see here exhibits the same three tunics, tunica interna, tunica media, tunica externa. The main difference is fewer elastic fibers and more connective tissue fibers. Okay, so tunica externa is where you have more connective tissue fiber. All right, so with that, let's talk about arteriole. Arteriole has all three layers. It's a smaller vessel. You're looking at endothelial cells, and then the basement membrane. You're talking about smooth muscle cells. Not a whole lot of smooth muscle because these are already narrower and smaller. And then these smooth muscle fibers are the ones that are very important in regulating the size of the lumen in the arteriole. Okay, this smooth muscle is supplied by autonomic nervous system, mainly the sympathetic system. Sympathetic system is the one that typically supplies blood vessels. Okay, except in a certain place where you see parasympathetic supply, almost all other blood vessels receive a sympathetic supply. All right, now let's talk about the vein and its histology. It also exhibits all three layers. You're talking about tunica intima, tunica media, tunica externa. What you see is that the muscle layer is less, right? Tunica media is thinner. Tunica externa with the connective tissue is a lot more than in the other types of vessels. All right. Generally, the way to think about it is that vein has a thinner wall, and it is distensible. That's the beauty of it. It can distend many times, the wall is such that it can expand. It can become a sac or a bag as it fills with blood. Okay, blood can pool in here easily, whereas in these other vessels, the blood doesn't pool. It has to keep moving. All right, so all three layers are present, but it is much thinner compared to the other vessels like the muscular artery or the elastic artery. Well, medium-sized vein, I wouldn't say, except for the size of the vein overall, it's still the same three tunics and the same kind of setup, okay? Now, one other thing that's shown here is the presence of valves. That's correct. Veins have valves, okay? Valves are structures that prevent backward flow of blood, and veins do need them, especially the veins in the parts of the body below the heart. That's where gravity pulls blood and keeps it in the lower parts of the body, and blood has to move against gravity, and that's where, as the blood moves towards the heart, the valves would close and prevent backward flow, and that is how you would keep moving blood from one part of the vessel to the next until it reaches the heart. Okay, these valves are fewer or absent from the veins that are in the upper parts of the body, where you do not have to worry about moving blood against gravity. Okay, good. With this, we will talk about the venule. Venule is where, again, you see all three tunics, and everything is much, much more thinner compared to the large vein or the medium vein. Now, let's talk about capillary bed. Here, the details are lacking. This is a capillary bed you're looking at, and this is a nice detail where you can talk about an arteriole here giving rise to capillaries in the capillary bed, and then here the fusion of the capillaries to become a venule. Okay, so there is a lot to be learned here. The histology is, first of all, just one layer of cells, endothelial cells, and a thin basement membrane. That's it. Okay, so tunica intima, endothelial cells, and basement membrane. No tunica media, no tunica externa. So that is its histology. And these are the smallest vessels. These are microscopic vessels. Okay, they go right next to the tissue, and they're the ones that allow exchange of substances between the blood and the interstitial fluid. All right, from the interstitial fluid, it reaches the tissue nearby. All right, so another important thing to notice is the presence of precapillary sphincters. These are smooth muscle structures that control the opening and the closing of the capillaries. All right, so depending upon the blood supply, the capillary sphincters, precapillary sphincters, by opening and closing, will allow the passage of blood through capillaries or not. So in here, what we can imagine is that these precapillary sphincters are situated in such a way that they block off blood flow to the rest of the capillary bed. So now the blood is moving from the arteriole through just this narrow portion of the capillary and reach the venule. Okay, so that is possible. And if there was more blood supply required, precapillary sphincters would change their position so the capillaries are open so blood can move in these capillary beds, okay? So the quicker way to move blood is from the arteriole directly into the venule via one short stretch of capillary, okay? This is what you call as thoroughfare channel, whereas here it's the capillary bed that we need to go through, we need to take the blood through. All right, arteriovenous anastomosis. This is another detail we can pick up. Anastomosis. Anastomosis is where you have additional connections or shortcuts, you may say. You see here, a branch is added directly from the arteriole into venule. Here, you can bypass the capillary completely and move blood from the arteriole into the venule. All right, so again, for quicker blood return, you may use this vessel than this vessel, you see. Anastomosis can be added. They could be between two arteries, two veins, or between an artery and a vein. So they can grow as needed, and that's what you're looking at in here. All right, so quite a bit of details in this slide. And now we are going to be talking about a cross-section of an artery and a vein. You already talked about the cross-section of an artery and a vein in the previous slide. You have more smooth muscle in the artery, so the wall is thicker and firmer and maintains its shape in a cross-section, whereas here in the cross-section of a vein, you see fewer muscle fibers, so the wall is thinner, the layer is thinner, and then there is more connective tissue. So, given all this, when you take a cross-section of a vein and an artery, what happens? The artery retains its shape, whereas the vein doesn't. And you can see how the arterial wall is thicker, and then the veins wall is not so thick. Then the lumen in the artery is round and smaller compared to the lumen of a vein. It's not even round. The way it collapses in its cross-section determines the shape of the lumen, all right? And then it's much wider compared to the lumen of a large artery. So those are some details. Those are some details of the histology of the blood vessels and the summary of the characteristic features of each kind of blood vessel is what we talk about next. Arteries, arterioles, capillaries, and veins are detailed here. First of all, arteries have the highest blood pressure. Okay, the pressure could be as high as 120 to 90 millimeter mercury, especially in the very beginning of the artery that's close to the heart, you see. And we already know that there are two types of arteries, elastic and muscular. Elastic arteries are the ones that serve as pressure reservoirs. So this is where they keep generating the pressure by their elastic nature, and that's why they're called pressure reservoirs. Now let's talk about arterioles. Arterioles are referred to as resistance vessels, major resistance site of the circulation. Why? They have a thick wall and a narrow lumen, and they're innervated by the sympathetic system. It is the control site for blood flow, and the pressure in here drops from about 93 to 37 millimeter mercury. So this is mean arterial pressure, which we will talk about later. That is 93 being the highest, close to the heart, all the way to 37, close to the capillary. Okay, arterioles have the narrow lumen, and they are referred to as resistance vessels. Very important for you to know. That is where the regulation of the lumen size is important. in generating the resistance as well as the blood pressure. Okay, very good. Now the capillaries. Capillaries, as you have already heard, are microscopic, and they have largest surface area. That is that the branching is so extensive that they create the largest surface area. So look at the surface area, anywhere from 500 to 700 square meters. Okay, they're called exchange vessels. Exchange vessels because they allow exchange of substances between the blood and the cells or tissues. So that's because of their thin wall and also because of the velocity with which blood flows. Blood flows very slowly here at the capillaries. Okay, so that is how exchange can occur between the blood and the tissues. And we talked about the presence of precapillary sphincter, which is basically smooth muscle that controls the opening and closing of the capillaries. And capillaries do not have any innervation, whereas arterioles are well innervated. We talked about that. Veins are the ones you refer to as capacitance vessels. So at this point, you probably notice that each vessel is given a nickname. Arteries are called pressure reservoirs. Arterioles are called resistance vessels. Capillaries are called exchange vessels. Veins are called capacitance vessels. What does capacitance vessel mean? Well, capacitance vessels basically keep blood within them. Okay, blood doesn't budge from here. It doesn't move out of the vein as easily as from the artery or the arteriole. Couple of reasons. One is the blood pressure in here is the lowest. All right, so there is not much force to push blood out of here towards the heart. The second reason is the wall is thin and can expand, and that is how blood can stay here. Low pressure and distension of the wall allows the accumulation of blood in large amounts within the vein. That's why they have the greatest amount of blood than any other compartment, not in the artery, not in the arteriole, not in the capillary, but mostly in the vein, not even in the heart, you see, mainly in the veins. How is that possible? Well, you know the amount of blood that's already in the heart. At any one time, the volume of blood ranges from about 55 milliliter to about 130 or 135 millimeter in each chamber. So that adds up to about 260 milliliters of blood, okay? Whereas in the veins, about 55-60% of blood is all sitting within the veins, okay? Especially in places like in the spleen and the liver. All right, now that's one detail we picked up. Highly distensible, which we had talked about earlier as well. The wall being thin, the lumen being wide, the vessel distends. And here I have shown 8 times more distensible than arteries. And we also talked about veins being equipped with valves, okay, to prevent backward flow of blood, especially in the lower parts of the body. The last thing we want to talk about, the blood vessel type or anatomy or histology, is the differences in the types of capillaries. All right, it's a beautiful illustration showing three kinds of capillaries. One kind that's shown here is continuous type. Continuous type basically means that the endothelial cells are arranged right next to one another, leaving very little space. That's why they're called continuous. These capillaries basically, I mean, they all have basically the same thing, endothelial cells in the basement membrane, and the endothelial cells here lack any space between these cells. Okay, these capillaries are typically found in the brain, in the skeletal muscle. This is where they go, deliver blood and collect blood and are done. The second type that you see here are called fenestrated. Fenestrated stands for porous. Here you're talking about porous walls, okay, meaning they have tiny spaces between the endothelial cells, okay. These spaces, the size of these spaces can vary, all right, depending upon the nature of the capillary. So what's the purpose of these capillaries? Why do they have these fenestrae is something you may think about. Well, two important examples we could think of are endocrine glands and kidneys. Okay, these are the two places where you find fenestrated capillaries. And in the endocrine gland, you probably imagined already, they are making hormones, and hormones have to get to blood. And how do they get to blood is where you think of fenestrated capillaries. So because of the porous nature of the capillary, hormone can get into the blood easily, and that is how it's transported. In the kidney, these capillaries are specifically in the nephron, in one part of the nephron called renal corpuscle. Glomerulus is the capillary network that consists of fenestrated capillaries. Okay, so blood moves through the glomerulus. Okay, as it moves through the glomerulus, particles that fit this space, the space of the fenestrae, escape. All right, so that is how it either helps with filtration or helps with the entry of hormones, etc. So these can be found around digestive structures, especially the small intestine where the absorption of nutrients occurs. Okay, so those are some examples of fenestrated capillaries. Let's look at the last type of capillary, but not the least. These are called sinusoids or discontinuous capillaries. As the name suggests, here the endothelial cells are far apart from one another. The gaps are much wider than in the fenestrated capillaries. These spaces are wider, incomplete basement membrane. And this kind of arrangement is important because you have some places where you need to move larger substances through the blood vessel wall, larger surfaces, larger molecules such as blood cells, proteins, you see. So that's where these vessels are important. Sinusoids are typically found in the liver, the spleen, and the bone marrow. Okay, this is where you're moving blood cells, red blood cells especially, into the tissue to be recycled. And the liver is where it makes a variety of proteins, and proteins have to be released into blood, and these capillaries are ideal for that type of function. Okay, so this is all you want to know about the anatomy, the histology, the classification of blood vessels, as well as the circuits We will now get into the different discussion, that is the physics of blood flow, okay, patterns of blood flow. So that would be our next video, video number 2 on vascular physiology. Thank you.